my talk is about Physome-based in silico research in the field of pharmacy and as well as medicine, which I have been doing so far. So in the meantime, two main approaches to develop in silico system, such as a deterministic and a stochastic model, will be introduced. So here's what I'm going to talk. First, I will introduce structure of the cardiac physio model, which we have developed so far. And second, application of cardiac physio, such as a drug cardiac toxicity evaluation and cardiac arrhythmia. So first, the structure of cardiac physio model. So the goal of the Physome is to develop a strategy and the methods for integrating vast amount of biological function information. So far, we have suggested a methodology to integrate the biological function and data as much as we can obtain in worldwide. Though there are still lack of data and the lack of mechanism which we have to reveal. Therefore, we have developed a cardiac physio model, particularly cardiac physio model in order to use to predict a specific biological function and drug responses and apply medical devices and the pharmaceutical industry. So among hierarchy of biological system from DNA to organism, this is a range of genomics and proteomics, which are study of structure and the function of gene and the protein respectively. Here, the range of the Physome, especially for cardiovascular system, is from between the part of protein and cellular level to organ and organism level. So microscale here is represented with a Markov model of subcellular molecular kinetics, which is mathematically formulated with several ordinary differential equation, which is called a lumped parameter model. Macroscale here can be represented with, for example, electrical conduction model, which is mathematically formulated with several partial differential equation, considering both time and the space at the point of continuum mechanics point of view. Here, the cardiac muscle is electrically excitable and also mechanically excitable. So we have to formulate it with electrophysiological point of view and also mechanical point of view. So in the microscale here, the ion channel is formulated based on the Markov model, which is based on the chemical reaction between state and the state. And this, so this can be formulated to whole different type of ion channel, ion pump, and exchanger. So if you integrate them all and incorporate them to the cellular membrane here, then you can simulate a cellular response in terms of electrophysiological point of view. For example, ionic movement across the membrane and the membrane potential, shape of the action potential, things like that. So single cell model is constructed based on the mathematically ordinary differential equation. And in the macro scale, we can make the electrical conduction model based on the continuum mechanics point of view. So we solve the conduction equation passively, then we incorporate active electrophysiological cellular model to the 3D model here. Then you will implement and simulate electrophysiological model of whole heart. in same manners, you can the electrical potential in the heart reflected to the body surface. Then you calculate conduction through media inside your body. Then you can calculate body surface potential. For example, cardioelectrocardiogram. And in the mechanical point of view, you will make the cross-bridge mechanism in a myofilament model is triggered by calcium signaling from the electrophysiological model, and you simulate cross-bridge model, and also in the macro scale, you will simulate based on the constitutive law and the stress equilibrium equations and under the hyperelasticity material property. Then you incorporate cross-bridge model into the mechanical hyperelastic model. Then you can simulate pumping heart, and which pump the blood, and the blood pumped through the artery. Then also the hemodynamics is coupled. So based on the Navier-Stokes equation, you will calculate and simulate hemodynamics as well. So this is a whole schematic of for the cardiac physium at the moment. So in the here 3D heart models contains electrophysiological cellular model, which contains subcellular molecular kinetics, including all kinds of membrane protein, as I said before, and intracellular protein being involved in the cellular electrophysiology. So normally, the 500,000 to 1 million cellular model is incorporated and distributed to the whole ventricles in order to obtain the reasonable response of ventricular electrical propagation. So this is the governing equation for the electrophysiology model of a single cell. And here's for electrical wave propagation through cardiac tissue, we adopted a biodomain model, which contains a reaction diffusion equation in two domains, such as intracellular part and also extracellular part, which is called interstitial space. So we calculate both of intracellular space and the interstitial space at the same time. So this is the reaction diffusion equation in interstitial space, and the second, this one is that in the intracellular domain. So left side is diffusion term in a passive electrical conduction, and the right side here explains a reaction term based on the single cell electrophysiology. Secondly, here the cardiac mechanics model, at the mechanical point of view, considered micro scale and also macro scales. In the micro scale, the molecular kinetics for the cross-bridge mechanism, which is formulated by employing a stochastic Markov model, considering troponin, tropomyosin, calcium signaling, and things like that in order to predict amount of active tension generation based from the myofilament. In the macro scale, here, 3D heart mechanical model is implemented by formulating with the equilibrium law and the constitutive law of hyperelastic material property for soft tissue model. So stress equilibrium equation and constitutive laws we calculate together, then you will get stress distribution, strain, and displacement, force, and active tension, and deformation of the heart. So this schematic represents how we implemented excitation and the contraction coupling of heart. So left part is electrical point, and the right part is mechanical component. Among two parts, of electrical circuit here. So this electrical circuit represent electrophysiological model of a single cell. So that is embedded, incorporated into each of the node here. Okay. So this electrical circuit model represent cellular electrophysiology. So here the resistance represent ionic channels, and the battery represent non-state potentials. Arrow here, current source represent ionic pump. So resistance, which explain the ionic channel, is not a constant. This is consists of several ordinary differential equations, which are formulated by experiment in the protein level. So in the right, the cross-bridge dynamics here, which is incorporated to the 3D cardiac mechanical model. This is also connected to the long-term model of a vascular system here, in order to give the boundary condition of intraventricular pressure to the ventricular wall. So here, so there are three physics. This is a Markov model of cross-bridge, and which is ordinary differential equation, and this is a continuum model of hyperelastic FEM model, and outside here, electric circuit is represent hemodynamic model. So these whole systems, we implement them all together, and we calculate them together, then we can get any physics of cardiac response, electrically and mechanically. So this gives you what we can obtain from the excitation and the contraction coupling simulation. We can obtain the action potential propagation through the cardiac tissue and the mechanical contraction as well. And all the responses in the microscale, subcellular level, such as states of all the protein subunits, all the concentration of electrolyte, and all the electrical current across the cell membrane. So this is the architecture of in-house code software in our labs, thanks to the Aroli, Marcel Linus. There's a pretty big international database from the in vitro assay and cellular and tissue experiment named the CellML. So there are a lot of data from the experiment in the CellML, which are fully open. We have developed the CellML code formatter, which convert CellML format of the mathematical model into C++ code, which is suitable to our simulation system, named CEP cell here. CEP cell system contains all kinds of cellular model, all kinds of mutation model as a plugin, and all kinds of drug effect, which have been published so far. So from this, whatever you choose among them, so you can computationally simulate and obtain the response in a microscale level, cellular level. So action potentials and all the current, the state of a protein. Whatever you are curious about single cell models, you can get from this system. And from this, also this CEP cell system can be also incorporated into the CEP 3D system here. And you can simulate computationally and obtain the response in the macro scale levels, like the organ level response, and the electrical propagation through the heart, and the virtual ECG waveform, and the body surface potential, like that. So for the validation of the virtual heart in cellular levels, action potential shape from simulation were compared with the experimentally measured one for both the ventricle and atrial cell. In the macro scale, we simulate and obtain the electrical propagation through the atrium, ventricle, reflected to the lung, reflected to the body surface. Okay? And the virtual 12-lead ECG waveform. So the personalized virtual heart, we obtained and used the geometrical model of heart from the specific patient and considering the placement and the location, direction of a heart inside a torso as well. Then we found that the virtual ECG waveform was quite similar to with the real ECG waveform, having only 5% error. So, so far I have explained the structure of cardiac Physome model, which we have already developed. From now, I'm going to deliver one of the application of cardiac Physome model, which is for evaluation of drug cardiac toxicity. So, in the background of that is evaluating the possibility of triggering cardiac toxicity of existing drug have limitations. So high risk drug causing cardiac arrhythmia, for example, have not been filtered out well during the new drug development stage. So this table is a list of drugs previously distributed in the market, but withdrawn from the market due to the tachyarrhythmia induction. So, and there are cases where the evaluation was wrong and cannot be distributed, even though it does not actually cause the cardiac arrhythmia. So, appropriate evaluation guidelines need to be prepared to accurately determine the drug heart toxicity. Therefore, the American FDA established the 4-stage assay named the SIPA project to solve these limitation and the problem and evaluate the drug safety. So the 4 stage of the SIPA model is like that here. So first branch is in vitro assessment of drug effect in the multiple ionic current, which has to be done in the protein level patch assay. Okay. Second branch is in silico computer modeling to predict the risk. This computer model have to get the data from the in vitro assay in the first branch, so which is being explained, and this part is our research topic. And the second branch is in vitro effect on the human stem cell derived ventricular cardiomyocyte in the tissue or cell level experiment has to be done. And the in vivo ECG biomarker in a phase 1 clinical trial has to be done to be accepted. So 4 of the branch, okay, part of the experiment has to be done for the drug cardiac toxicity estimation. So, as I said, the cardiac Physome model can be mainly used for the stage 2 here, which apply data obtained from the stage 1 into the model. So Physome model obtained in vivo data from stage 1, as I said, and computationally simulate and provide a cellular response and organ response as well. So from the PET experimental data, in silico simulation in cell level and organ level. So we simulate the shape morphology of action potential shape in a single cell level, and also we simulate the electrocardiogram. Both are very, very important for estimating the cardiac toxicity from the drug. To explain in more detail for each step, we first obtained the 2000 set of IC50 and the Hill coefficient, which is from the experiment using bootstrap, which is a way for uncertainty quantification of the raw in vitro data. This part, thanks to the Ali, has done create the uncertainty quantification algorithm, implemented it, and we use it because of due to the limitation of experimental condition. We need to get the more reliable data from the experiment. So we did this UQ solution. And 2000 set of the IC50 and the Hill coefficient is calculated in the way are used as input for the in silico cell model according to the drug concentration here. Then we get cellular response under the drug. So we use the cellular response data from the simulation and classify drug toxicity by applying the machine learning approach from the cellular data, which is calculated from the simulation. So FDA research group provided QNet and QInward as a biomarker for drug toxicity evaluation with a logistic regression, while we provide more sophisticated algorithm such as artificial neural network and CNN, and we compare the performance between the FDA group and our group. So we got we did several machine learning algorithms, logistic regression model, and ANN and CNN. We trained them and we tested them with already known drug. So we got this scores. This system classified high risk drug group and the low risk drug group, and also intermediate risk group. So we got this this score, which is pretty good. And this is published to the journals under the when we use the artificial neural net. So we provide better performance than that FDA provided here. So logistic regression, which is done from the FDA, and the ANN from us. So we have the better score than the FDA group. So this is one of the reasons. This is the whole schematic. Explain the in vitro, in silico, and the artificial intelligence integrated platform. So experimental data and uncertainty quantification and single cell simulation and the machine learning together. Then once you do in vitro assay, then you will get drug toxicity prediction. Okay, so from now, I will introduce another application of a cardiac Physome model related to the cardiac arrhythmia. Yeah. So yeah, I will explain briefly here. So this is about prediction of cardiac mechanical performance from electrical feature during the ventricular tachyarrhythmia simulation using the machine learning algorithm. So this is also we do in silico deterministic deterministic simulation, and we get three-dimensional data, and we extract a feature from the data, and we use them to the machine learning. Then we predict mechanical performance. And this is about the computational prediction of effect of mutation named D172N KCNJ2 mutation on the ventricular pumping during sinus rhythm and the re-entry arrhythmia condition. And this one is about the virtual defibrillation simulation, which provide optimal defibrillating protocol. So we induce the tachyarrhythmia here, and we will apply the defibrillation virtually with a specific protocol here. Then we know we predict whether this protocol is successfully defibrillate the heart or not. So this simulator is used for the predict and obtain the optimal defibrillating protocol as well in the three-dimensional model. Okay, so yeah, so far I have introduced the structure of a Physio model first, and application of Physio model in the field of pharmaceutical and medicine, cardiac arrhythmia. Now, I will briefly introduce machine learning research for the cardiovascular system, such as continuous blood pressure measurement and cardiac arrhythmia classification. So this is for the continuous blood pressure estimation using simultaneous use of ECG electrocardiogram and also PPG, photoplasmogram waveform. ECG has the information of how membrane potential propagate through the cardiac tissue. What's the action potential shape like? When to study, the depolarize and repolarize, and when to finish them, things like that. And the PPG has the information of what's the blood pulse shape like, and the time for the pulse transit time, things like that. From the both of ECG and PPG signal, we are able to extract important feature for estimating blood pressure, such as pulse transit time, pulse wave velocity, and stiffness of artery, things like that. However, we may not be able to know all the important feature for that. Okay, in the case, so we don't trust all the feature we can obtain, but we expect there are more features which are available. So we use the raw data, shape of the ECG waveform and also PPG waveform for the input to the CNN model. So we will rely on the CNN feature extraction protocol. So we give the difference of ECG waveform and PPG waveform, then which can have both of the feature from ECG and the PPG. So we use, as I said, we use the CNN, and also we adopted the bidirectional LSTM together. So as a result, we have estimated systolic and diastolic blood pressure continuously with quite good score using the standard scoring system. We got here, so this is our models, SBP and the DBP. It has the small standard deviations, and we got the A score. And last, this one is the cardiac arrhythmia classification. Concept is like from 12-lead ECG waveform, we classify 9 different type of cardiac arrhythmia. cardiac arrhythmia has very many different type of cardiac arrhythmia. So important 9 different cardiac arrhythmia was classified by our machine learning algorithm. This workflow explained, first we got ECG signal, and we have heart peak detection, and we will get from heart peak to heart peak after 3 cycle. Then we get that 3 cycle signal as a group, segmented. Then we clustered several samples and as to one, and we use that final 3 cycle signal, process the frequency feature map. Then we put it to the 2-dimensional CNN and classified 9 different arrhythmia. It was just we did this one several years ago from the Computing in Cardiology Challenge. So this is the result. So yellow is represent just case which are quite good. So atrial fibrillation and ABP, left branch bundle block, and the right branch bundle block. The 4 of the arrhythmia was classified quite good among the 9 arrhythmia, and whole weighted F1 score was 0.78. So it has been published in the journal. Yep, I think I introduced all of what we created so far, and I want to thank to all the member of a computational medicine lab here. And we got 5 PhD course student and 5 master student course. I thank to my members here.